Hello students, in today's class we are going to learn about post-translational modifications. So these are the modification or changes that take place in the proteins once they are synthesized. The modifications take place in prokaryotic as well as eukaryotic proteins. So post-translational modification majorly involves folding and processing of the newly synthesized polypeptide chain. Uh, so folding reaction is guided by several interactions such as hydrogen bonds, van der Waals interaction, ionic and hydrophobic interactions. Many polypeptide chains also undergo additional processing apart from folding, which are known as the post-translational modification. So we will be looking at few examples of different types of modifications in the following slides. So first of all, we have modifications of amino terminal and carboxy terminal amino acids. So in case of many polypeptide chains, uh, the first amino acid, which is usually formilated methionine residue in case of bacteria. So majorly the formyl group as well as the initiating methionine residue and sometimes also the carboxy terminal amino acids are enzymatically removed and they are not present in the final functional protein structure. Second category of modifications are loss of signal sequences. So signal sequences are crucial for targeting a protein to their final destination or their location in the cell where they function. For example, in case of eukaryotes, you have these different organelles uh, such as nucleus and so many different organelles, and the proteins have to be transported from uh, the location of protein synthesis, that is cytosol, to different organelles. So this uh, proteins, many of the almost all proteins, they have some or the other signal sequence. For example, once the proteins are synthesized, they are transported from the cytosol to the nucleus. So these protein contains a signal sequence which is known as nucleus localization sequence. So many a times these sequences that are signal sequences, they are not present once in the final structure of the protein. And once the protein has been transported, they are enzymatically cleaved off. This is also true in case of prokaryotic proteins where they are transported to different locations such as the periplasm or the inner membrane or the outer membrane and so. Then the next modification is of individual amino acids. So these type of modifications such as those catalyzed by enzymes, kinases and phosphatases, they are basically associated with addition and removal of phosphate groups on specific amino acids. These type of phosphorylation and dephosphorylation reactions are quite common in case of bacterial kinases, and they play an important regulatory function in bacterial system. So here we have an example of like a pair of proteins that often function together. The first protein is known as a sensor kinase protein, which has an histidine residue. So the sensor kinase component can identify or sense some kind of a signal, for example, presence of a particular nutrient. And once the signal is received, then its histidine residue undergoes an autophosphorylation reaction, wherein the histidine amino acid is phosphorylated by making use of energy from ATP. And This leads to activation of the sensor kinase subunit. Once activated, then it acts upon the downstream protein, which is generally a response regulator. So the two proteins, the sensor kinase and the response regulator, they work in combination. So what happens once a sensor kinase is being activated by autophosphorylation, it can transfer its phosphate group to another amino acid, generally an aspartate amino acid present on the regulatory protein, and by a reaction which is known as a phosphotransfer reaction. So the aspartate residue is now phosphorylated, and this leads to activation of the response regulator protein, or you can simply call it as a regulator protein. It's sometimes known as response regulator protein because now this is the one that will show the response by, for example, binding to the downstream promoter regions of the downstream genes that it is regulating. Once it binds to the promoter regions or near the promoter regions, it will allow or increase the expression of the downstream gene so that the proteins are expressed, and then they can respond, show any kind of response, for example, chemotaxis, that is movement towards the nutrient that is being identified as a signal by the sensor. Okay, so these kind of two component regulatory systems are very common in bacteria and are used for regulation of many different types of processes. The next category of modification are protein glycosylation. So, like this, in this modification, you have formation of glycoproteins basically. So, there are two common types of glycosylation reaction named as O-glycosylation and N-glycosylation. So, we know about amino acid residues. There are certain amino acids such as amino acid serine that contains a OH group or a hydroxyl group present as in its R group. So in an O-glycosylation reaction, you will have replacement of the hydrogen of the hydroxyl group and attachment of the sugar to the oxygen of the hydroxyl group. And hence it is known as O-glycosylated protein. At other times, you will have certain amino acids such as threonine that contains amino group as a part of its R group, and the sugars are attached to the nitrogen of the amino group, and hence the reaction is known as N-glycosylation. Okay, so this type of glycosylation reaction is found very common in proteins such as pillin proteins, which are proteins that are involved in formation of pili structures, which are involved in attachment, and also found in flagellin proteins, which are building blocks of the flagella that are involved in bacterial swimming motility. So these modifications have an important role in adhesion and stabilization of the protein against proteolysis, and also uh, by evasion of the host immune system. The next category of modification is addition of prosthetic group. So there are several proteins which contain protein component that is synthesized by protein synthesis. And in addition to the protein component, they also contain a non-protein part, which is important for their activity. The non-protein part that is present in functional proteins is known as its prosthetic group. There are several examples of proteins with prosthetic groups. For example, pyridoxine are common proteins that contain iron and sulfur as their prosthetic groups. Pyridoxines are important components of the electron transport chain in aerobic as well as in anaerobic respiratory processes. Uh, Many of these pyridoxines are also component of electron transport chains. Here are mentioned some of the anaerobic electron transport chains such as nitrate, nitrite, and sulfate reduction or respiration process. Then we have proteolytic processing. So there are many proteins which are initially synthesized as large inactive forms, which are known as precursors. And then these are proteolytically cleaved into smaller and active forms. This kind of processing is quite common in bacterial toxins, which achieve their high level of potency by delivery of the catalytic active polypeptide fragment of the toxin to the eukaryotic target cell cytosol. So it is commonly observed in case of diphtheria toxin or anthrax toxins, which are uh, which are proteolytically cleaved and then they are converted into uh, enzymatically active and potent toxins. This type of cleavage, proteolytic cleavage, is also seen in case of some of the eukaryotic proteins such as insulin. So insulin protein, when it is initially synthesized, it has three chains or three parts to it. The A chain, the B chain, and the central connecting chain that is not shown over here, which is known as the C chain. So C chain, basically the function of C chain is to allow the folding of the chain so that the A and the B chain can come in close proximity with each other and form the bonds. Once the bonds are formed, then the C chain is cleaved off and the active insulin protein consisting of only the A and the B chains is left behind for acting. Last type of modification is formation of disulfide bonds. So the amino acid that is involved in disulfide bond formation is cysteine amino acid because it has presence of SH groups or sulfhydryl groups present. So these type of bonds are sometimes important in formation of a final functional active protein molecule. In some enzymes such as alkaline phosphatase from E. coli, which is a homodimer, um, so these disulfide bonds play an important role in formation of these homodimers, wherein the dimeric form of the protein is fully active and enzymatic form. So that's it about a few examples of eukaryotic and prokaryotic post-translational modifications. Kindly like and share if you like the videos and subscribe to get direct update on the uploaded videos. Thank you.